Can't find my microphone. Oh, it's on. Hey everybody, long time no see. I saw some familiar faces as you were popping up. A couple of you I don't recognize. I'm Shannon Egley. Welcome to Microscopy 101. We have 3 intro videos for your kit to help you get to know microscope a little bit, learn some cool skills, and then try some things all on your own. So 101 is we're going to play with things, open up the scope, talk a little bit, play with things that you don't really have to have any special skills to be able to do, just so you know how to use your microscope. 102 is using or developing particular skills like how to make a slide, how to stain specimens, how to make a permanent mount. So taking a fly or something and making it a permanent fixture on a slide. And then applying some of those skills in part 3 to make your own experiments. So I'll give you the topic, share with you a little bit about it, give you a slide and let you figure out how to use it. Of course, I'll be there with you all the way, but I just want to watch you go through and be able to build slides on your own and experiment on your own. So you can grab things around the house, whether it's something out of the fridge and you want to look at it up close, like some yogurt or it seems weird, maybe a slice of ham or turkey where you can see the cells inside of it. Hopefully you see the cells. If you can't see the cells inside of it, then you might not want to eat it because it's not so real. But this is Jump Simulations microscopy class with OSF and University of Illinois College of Medicine helping. And again, I'm Shannon Egley. I'm the anatomical coordinator at JUMP and the clinical associate of anatomy at University of Illinois. So what's kind of fun is really looking forward to the microscope. It's a tool that I thought when I was in high school was probably the first time I used a microscope that I'd probably never use again because I wasn't really into science or biology. And then I used it again in college and grad school, and I use it now too. I love just finding things randomly, whether it's something around my house or I grabbed some leaves actually on the way to work yesterday off a tree that I thought was really cool to check them out and see how they're built. But hopefully you get into it. You'll find, I think, through the course that it's not just science people or people that are interested in medicine or anything like that that have to use microscope. It's anything. If you like jewels, diamonds, rubies, gemologists use microscopes. Geologists that look at rocks, they use microscopes. I can just go on and on. Forensics, pharmacy, all of those. I look at my book. But you should have, well, first of all, hopefully you all opened your microscope and made sure that the batteries went in and it worked okay. It's like I can't really see anybody. If you're having any problems with your microscope, type in the chat. I'll open that up right now. If you have any random questions, you can pop in the chat too. But you should have this book. So if you don't have a black binder, then look in your mailbox because it's in your mailbox right now. And just let me know you're going to your mailbox so I can install. So inside here are the first 2, 101 and 102, 103 and 201, 210, all of those other ones are coming to you. I think they're actually being posted on Monday to go live so that you can print the PDF and insert them into this. This is nice because you can flip around in it. If you see a page that you think is really helpful, you can take that page out, move it to another area of the book, just like a recipe book. So if you have a section of your recipe book that you have favorite recipes, you can take that page out, put it back in the favorite recipes. Maybe get a tab sticker or something and mark that. Make sure you have the book. I'm going to walk through the first couple pages with you. There you go. So it should look like that on the front. And again, the first section is 101. And it's, oops, I'm on 102. Let's say 101. There we go. Whoop. Let's see. So 101, seeing is believing. I say that because there are a lot of things you're going to see that you've never seen before, unless you use microscopes kind of regularly. So keep an open mind, look at all the cool stuff, and then hopefully you'll explore with all the stuff that's around your house. flipping to the second page. We're getting used to all these little windows. Some of you have seen this before because I talk about it every time, and for the same reason, it's that you should really think like a scientist. You were born to be a scientist, whether you go into, well, I'm thinking of all scientists' jobs now, but engineering, if you go into architecture, doesn't matter what you're going to go into. If you don't think out of the box, you'll be stuck in the same box that everybody else has been in forever. You were born to think out of the box. You were born not knowing how to navigate around the world, not knowing how to use your brain, not knowing how to use your fingers, your eyes, or anything, and you had to learn really fast how to use all of those. When you were a baby, you could only see about 30 centimeters in front of you, so a little over a foot. That's as far as you can see clearly. So when someone's back away from you, you're just a blur to that baby. And they came towards you, and they became more clear. Your microscope is going to do the same thing for the microscopic world. There are things you're looking at right now that you think are clean, or there's nothing on them, or there's nothing really to be seen. But with the microscope, you can see all this new world that you've never thought about before. When you think about how much you're magnifying, if you take 1 teaspoon of dirt, just a little teaspoon, and you look at all the microbes in there are thousands of them in that little tiny teaspoon of dirt. And just imagine all the dirt around your house, not on the inside, hopefully, but on the outside and around. They're everywhere. So as you're going through this, if you have questions, let me know. If you have an open mind, you're going to have a lot of fun. I put some QR codes down here that you can explore. This is actually a QR code about babies that do think like scientists and how babies do a kind of math that they weren't even taught to do from the time of birth. So it's fun to watch. All right, second, 6 steps of the scientific method. And the scientific method's been around forever, for as long as we've been thinking. We just didn't always have it in steps. It's because this is how we change our habits or change the way that we look at the world. So we observe something. If it's not the way we want it, or we don't understand it, then we question it. We wonder, why does it work like that? Why does it have to be like that? We build a hypothesis. What can we do to change it? What can we do to prove that we're thinking correctly and maybe those people aren't, or maybe I'm not understanding it right? So your hypothesis is, what do you really believe? And then you experiment, test that thing. So if you're seeing it happen one way, change it, make it different, experiment with it, and see what happens when it changes. If I want to lose 10 pounds, my experiment might be, well, if I stop eating chocolate cake every night, I'll probably lose weight. That's not a hypothesis. If I stop eating chocolate cake every night, I will lose weight. So it's a definite statement. I experiment, I suffer through it, I don't eat chocolate cake for a week and see what happens. Maybe I lose a ½ pound or a pound. Then I've proven that it's the chocolate cake that's doing it to me. I should probably back off of it. And then I reorganize my question and go at it again. Well, I only lost a pound. I should probably try to lose a little more. So what if I cut out soda for a week? Maybe I lose another quarter of a pound. Or what if I cut out this and I lose another quarter of a pound? So things you may not have thought of being a scientific method are actually around you all the time. Anytime you question something that you see and you want to make it different or change it or get to understand it better, that's you applying the scientific method. Okay. So let's say maybe I observed that babies don't see up close, right? Or they don't see far away, they only see up close. And I want to be like that. Let's build an experiment right now. What could I do to experience life like a baby? What's it like to see faces from a foot away? I think that it would be scary. That's my hypothesis. I think it would be scary to only understand or recognize faces when they're closer than 1 foot away. So how can I do that? I take a Ziploc bag. Ziploc bag came with your kit. It's probably in the white envelope that says 101. Just like that. See 101 in the top corner. There should be 2 of them. And I forgot to mention this, but you'll definitely want some water, not to drink, but for experiments. The only thing I couldn't ship was the water, so, or get to you like that. So if you want to get a cup of water, while you're in there, get the cup of water. If you have a cylinder glass, that's ideal. And I think that I can build, forgot to turn something off. Sorry, everyone, for a second. Fixed. Okay. And now I've walked myself out of my picture. There it is, back. See, I bought you some time to get some water. All's perfect. So take your water, your Ziploc bag, and you're going to get this pretty full. If you can leave a little bit of space to zip it, that's ideal, but I'm just going to zip it over this water glass. I feel like I'm doing a magic trick. All right, it's all zipped. So you've built your first magnifying lens for the class. Right, it's not a perfect glass lens, doesn't do crazy magnification, but it will let you do this. I did it again. There it is. Wow, my whole world turned oompa loopy. So here's a lens. It's a basic water lens, nice and round, flat. Some of you dissected eyeballs with me, I know that. So you remember looking inside the eye, it's nice and round. The lens was like a ball. Baby's lens is like a ball, nice and round. They see things up close like this. You see the writing magnified? But if I flatten out the lens, no magnification, right? Well, barely. Only the rounded part's doing it. So what if my experiment said, I can have baby vision for a week? All I need to do is put a pair of glasses on. They can be regular glasses with no special vision, and this taped on the inside of it. It's crazy. So I can see everything. You can see my skeleton in the corner. It's all blurry, real blurry. So if I want to see something clearly, just look really close. And that's nice. I can read that. But I have to be super close, just like a baby. Don't just do this experiment and wander around the house like that. I don't want you falling and getting hurt. But you can build experiments, test things. I remember when I was in grad school, people were testing what it was like to have a neuropathy, which is a pain out of your fingers or your toes or your extremities. Actually, it's pretty much pain anywhere. And so what they did is, for the pain, they took some kind of pointy rocks, put them in their hand and put tight gloves on, and they put 3 tight gloves on. So they had the sharp rocks, not to cut themselves, but just that irritation, with tight gloves. So their fingertips were kind of tingly, but they had pain and shooting in their palms, and they wore that for a couple hours just to get that experience. So they were experimenting, wanting to find out what it's like to feel like somebody that's having neuropathy. So be creative, think out of the box. It was a really fun experience, and I actually forgot about it until now. Think like a scientist, use the 6 steps. This QR code is actually about a scientist. It was a doctor that he noticed patterns. He noticed that people with stinky breath got ulcers a lot. So he thought maybe there's some relationship. He also noticed that when he took a sample of their stomach acid, there was something moving in it, and it was bacteria. And all the other doctors he talked to said that's impossible. The stomach's too acidic. There's no way that bacteria can grow and live in there. It's just not possible. So he did something you really should never do. He took a sample of the bacteria and ate it. And he waited, and within a couple months, he started getting really bad breath, and he started getting stomach ulcers. And how do you get rid of a bacteria in your body? Take an antibiotic. So he took an antibiotic, fixed him, his ulcers went away, his stinky breath went away, and he discovered that it was actually this bacteria called Helicobacter pylori that was causing bad breath, halitosis, and stomach ulcers, when other scientists and doctors were thinking, it was incurable. And he cured about 90% of the people that had stomach ulcer, bad breath combinations just by giving them an antibiotic. So it's sometimes just the little observations and asking yourself, what could it be? What can I do? What can I test? Right? 3 tools you need. The first one is your brain. So have the right tools. You need to be able to flex your brain like you flex muscles. The more you flex them, the bigger they get. Your brain's the same way. Ask questions, lots and lots of questions. Thomas Edison had a quota, and they call it the Edison quota, where he asked a certain number of questions and came up with a certain number of possible inventions in a month period. That was his quota. He had to do every month. And what people find is if they try and do that, do the Edison quota, you might come up with 1 or 2 ideas now, and maybe a couple more ideas tomorrow, and then maybe 3 or 4 the next day, and then maybe like 10 or 11 the next day. But the more you use your brain and push it to ask why, the more questions your brain will generate. We were like that when we were 2 years old. We started asking why about everything, and then our parents were like, because I said so, and we quit asking questions like that. But it's time, you know, as high schoolers, start asking it again, because you're looking at career paths or things you want to do for your whole life. So ask why. Why do people do what they do? Why would I want to do what I do? So number 1 tool is your brain. The next thing here is not a tool, but it's something that's really important before we open up the scope: how to use your microscope safely. It's not a dangerous tool. It's a precision tool, and it can do a lot of things. One thing it can also do is it can cut you. So you want to make sure you're careful. Another thing that you can do to it is you can bust it apart, and you don't want to do that either. So you're working with glass slides, and you know, if you have a nice stable tabletop, it doesn't seem so bad, but a small drop for that little thin glass slide could shatter it into a ton of pieces. So you want to make sure, I'm trying to flip my page again. There it is. You want to make sure that, number 1, you have something near you that you can put broken glass in. So whether it's a cup like this, or my recommendation is just like a pickle jar, a used pickle jar. Keep it on your desk or your area. If you break something, use some paper, you know, like scoop it up with the papers and dump it in the jar. Try not to handle it with your fingers, because that thin glass can potentially be like a razor. So that's my piece of advice, and I would make a list of things you might want to keep around. So number 1 would be a sharps container, is what I would call it. Number 2 is a trash can. So oops, here, always within reach, so that if you spill something, you can clean it up, throw it away right away. If you have something you were experimenting with and you don't want to ever look at again, right into the trash. Right? And the third thing are paper towels, because if you're working with water or stains or anything like that, you want to be able to clean it up fast. So those are 3 things you really want to have handy all the time: something to keep sharps in, a trash can, and something to clean up with right away. As far as safety for the microscope, when you hold your microscope, you'll always hold it in 2 places. Here's the arm, here's the base. The arm is where you carry it up here. The base is just support. If you have to let go, make sure that you're like leaning against your body or something so it doesn't fall. We'll talk about all the different parts later, but the reason you want to hold it like this is in case something's loose, you don't drop the base off, break the wiring. You always have stability, so you don't tilt it all over the place. Right? Next, work within shouting distance of an adult. If you are working with the glass, I mean, you're responsible people. I remember a lot of you, but if you slip and you cut yourself and you're bleeding a lot, you want to make sure somebody can come help you. So Band-Aids would also be a good thing to have. Next one, never eat or drink near the microscopes. Eating and drinking in the same room is fine. Like my coffee, keep it way away from the microscope, so if it does spill, it spills on things that aren't going to get broken, for one thing. And number 2 is if you're working with something you don't know about, like you gathered some pond water from a stinky pond, you never know what's in that. There's something causing the stink, right? So you don't want to be eating and drinking or working with these things that could potentially make you sick, right? Next, handle chemicals and specimens with care. If using potentially dangerous or infective agents, wear gloves, glasses, and mask. You didn't get gloves, glasses, or mask with this kit because we're not working with anything that's going to hurt you. But if you want to experiment on your own and come up with some really interesting things, definitely make sure you're wearing your mask so you don't splash it or inhale anything. Gloves, so that if you do get something on your hands, number 1 line of defense is peeling the glove off. Number 2 is washing your hands quickly. And the same thing with goggles. If you're working with something that splashes, you don't want it in your eye. And if you need those things, Jump has they call it a sibling kit that has apron, gloves, glasses, mask, all of that for dissection. You can just, I think they're only a couple bucks. Right, 2 hands on the arm of the base at all times. I already said that right away because I want your microscope protected. Keep the scope covered when it's not in use. So mine, even when it's in the case, it's covered with a vinyl cover. And when I take it out, oops. Yeah. So in my defense, I just finished another class, but make sure it's always on. If something happens to this, just take if you have to, like a grocery sack and put over it, or a 1-gallon Ziploc bag would work too. All right, always start with this stage furthest away from you. So there's a lot of split decisions on this, or split opinions, I mean, on this. This is the stage, the black thing. So the reason that people say go to an extreme or another is for one of two reasons. One is because when you're turning the knobs, it's easier if you're in extreme to go the opposite direction only. The other thing is that when you're on the longest lens, this is called the high power objective, and it's all the way down, it actually sits really close to the stage. So if you have a piece of glass slide on here and you start cranking away and you're not paying attention, you'll break the slide. So a lot of people say if you just go ahead and start like closest, you can only work down and you're always safe. In my opinion, I like starting low and slowly working up. I mean, it's no race. I'm not trying to get anywhere fast. Right. So always start in extreme where the stage is supposed to be. And then never take the microscope apart. I'll show you how to take some of the things apart to clean it later. But in general, leave all the objectives and the lenses where they're at, unless like this one comes out to pack it away and you put a cover back over it. Don't open it up on the inside. It's a precision tool. It's just like it's like a saw, a power saw for somebody that enjoys working on wood, or a nice car for somebody that enjoys driving. It's a precision tool, and it may not seem like it's that inexpensive, or it may not seem expensive, but at the same time, it's a very quality tool. It has glass lenses on it, so they can use the scratch. It has a metal body, so if you drop it, you could potentially break it. Just take care of it. So it's not heavy metal, it's thin metal body. And then the last one's really important. That's why it's in capitals. Only use a lens cloth when you're cleaning it. You have a pack of lens cloths that came with your set, your microscope, like this. The reason it says only use the lens cloth is because this is 100% grit free. If you use something like, if you had science class and they give you chem wipes, like this, these are 80% grit free. So there's still potentially 20% grit, which makes this seem like sandpaper on a lens. So it's really bad to use these. Don't use them. Don't use a Kleenex. Don't use your shirt. When you're using anything on the lenses on the microscope, doesn't matter if it's the eyepiece, ocular, or the objective, always use lens paper. If you need something wet to clean it with, you can go to, I got this at Walmart, I think in the checkout line, but Zeiss is well known because they're one of the oldest microscope companies in the world, but they have a really good lens cleaner meant for glasses or cameras. If you don't have this, you can take Windex and dilute Windex 50%. So it's 50% Windex and distilled water that you put in it. Make sure it's not tap water because there are a lot of minerals in there that could potentially cause spots and things on your lens. So always use lens paper. That little QR on the bottom is if you want to learn more about how to clean and take care of your microscope. It's a reference to a science supply company and a video training they did on taking care of your microscope. All right, next tool, next page, your lab. So kind of jumped over here. So tool number 2. This is a really cool set. You've got pretty much everything you need. All you have to do is get refills on some things. We do a lot of experimenting. But I tried to make sure that everything that you got is a complete microscope lab. There's nothing you have to have that's not here. Let's go through it real quick. You should have, I know you did, otherwise you wouldn't be listening to this, but you got the box with the scope in it. In the top of the box, you have a little box with all of the, well, not all of, a lot of the individual pieces. I actually got a toolbox from a local tool company. It's a 19-inch toolbox. I just made sure the measurements were big enough to hold the microscope. I like it because I can put all the tools on the top. Open this up. Inside, I can put the microscope. I just put some, when I ordered some products online, they came with the big air bubble packing things. So the microscope lays right down in it. And then I have another tray for all the random things that I like to look at under the microscope. So I have some diamonds in there, some shredded up money. Actually, serious. Cut up can. Things I really wanted to look at closely under the microscope. And hopefully you want to do the same things too. A packet of sports feed crystals. very cool. But it's great because all of it goes right into that box. I think I got it on sale for $6 for that big plastic box. And if I want to, I was gonna say if I want to keep my little brother, but he's not so little anymore. If I want to keep one of my kids out of the box, I could just put a lock on the front of it. And then with the padding, I feel a little bit safer that it's not gonna fall and break. Okay, so you got your set in front of you. Hopefully you tested to make sure the batteries are working and the lights are working and everything on the microscope. So that's part number one. Let's do, I want to be able to see your desktop, or I want you to be able to see my desktop. Like this. So we'll go through and check them off one at a time. You have the microscope. You have a lens set. These are called Barlow lenses. They're special lenses because they let you adjust, which most lenses don't. And we'll talk about that. We'll actually have a session on that later. But you have one that says WF10X, which means wide field. So your normal field of view that you can see, the little circle that you're looking at, is a little bit wider with this lens, and it magnifies automatically by 10 times. So you could actually use it like a magnifying glass and put it down and be able to read print with it. There's the Barlow tunnel or tube. It has a small lens at the other end. And you have a 20X. I'm not even going to take it out. You can see there also says WF 20X because this is 20 times magnification, so a little bit more strong, a little bit stronger than the others. You have a phone mount, which I thought was pretty awesome because I actually bought a camera so that I could record these, and I found that just having the phone mount and putting an old broken phone, so the screen's completely shattered on this one. There it goes. By mounting it, it works great as a microscope camera. In fact, I can just pop it right on. You don't have to just look at me the whole time. And I'll show you how to set that up in just a little while. Phone mount. What's also nice is that you don't have to hook it to your computer like this. You can just hook it up, watch the screen. You can take a picture when you think it's cool. You can focus or zoom in on whatever you're looking for, and then take that photo you just took and put it on Google Images, and Google Images will tell you, usually, I haven't had any problems with it, but it'll tell you what you're looking at. So even if I don't know what bacteria I'm looking at, I'm a little confused, it'll give me hints or tell me exactly what I'm looking at, which is really nice. It's not as easy as that with bacteria because you have to do other tests. But if I'm looking at some kind of an insect, I've actually done that a couple times, and I send it to Google, and Google knows everything. I swear, it's crazy. So phone mount, prepared slide set. You actually have two sets. You have, these fit in the toolbox too, but I just didn't put them back. The wood box. Careful when you open it. So I write on it. You can see I wrote top. When you open it, there are little subtle clues that tell you what the top is. The top is thinner, wider. This clips up, but it never fails. I'm talking to somebody or whatever, and I wasn't paying attention, and I'll open it up and dump all my slides. I just did that less than a week ago. The foam stays in because you don't want this tightly packed. If it drops, then all that force will go into the glass and shatter them. Here's your list of everything that comes in this. And we'll actually end up through all the sessions. We'll go through all these items. Plus, you'll be building the same number or more slides on your own. So that's the first prepared set. The second prepared set is a small white one. There it is. Small white box. Wrote top on this too. I don't like making dumb mistakes. I make plenty of mistakes, but I don't want to feel stupid opening up a slide box upside down. So this one has 5 prepared glass slides. It has 10 plastic slides. I'll show you how to use those. But I also give you transparency paper so that you can cut these to the same size. It doesn't have to be perfectly the same size as a slide, but you can cut these so that you can use them as temporary slides for wet mount where you're just putting drops of water with your specimen in it. You can actually build something that's supposed to be permanent, but keep in mind this is flexible, so if you bend it, it'll crack and fall apart. But it's nice because you can do a lot of experiments with this and you don't have to spend $1 on a glass slide for every experiment. All right, blank slides, they are in the plastic box. And you'll actually get some more blank glass slides throughout the add-on courses that you're already taking care of, and they're going to be coming your way. I think Monday is when you're getting access to them. Phone mount, prepared slides, blank slides, slide covers. That's the little white box. Not one we just went through, but this one. Only open this when you need to, because these will fall over the place and you have to be really careful and slow opening it. It has little labels that you can write on. It also has the very, very thin glass, which if you think cutting yourself with a razor is bad, if you cut yourself with that thin glass, it's like worse than a razor, because number one, you never know if any of the broken pieces were still on there when it went into you. So again, empty pickle jars are really good for things like broken cover slips or broken slides. Transparency paper, I already said. Ring stickers. Might have this one, you might have the other one, but the ring stickers are perfect because when you're making a slide, they build a well. A well is an isolation area. I can get that out. There we go. So if I cut out the size of a slide and put this right in the middle, now I can put a drop of water or a powdered substance or something in there, and it stays inside the well. It doesn't go anywhere. So I don't have to worry about it. It's also easy to find the material too, because you look for that big ring and then just go into the inside of it. Okay. Ring stickers. You should have 6 vials. So you should have 2 that have stains in them, a blue one and a brown one. If they're not labeled, it's easy to figure out because you just take and shake it, look at it upside down, and if it looks blue, it's called methylene blue for staining proteins. You shake it around, it looks kind of brown. It's iodine, which you've probably heard of before for cleaning wounds or cleaning skin before a surgery. But that's really good for staining carbohydrates, and I'll show you how to use those later. You have sea salt and, oops, there it is, brine shrimp eggs, so you can build a small hatchery. That's what this is for. So it looks like a petri dish. You can use it to catch specimens. You can also grow your brine shrimp in here, but it's a saltwater creature, so you have to have salt in the water. Yeast, you have packets of sugar too. Oops, that's still the wrong one. So yeast eats sugar, and it's fun to do experiments with them because they're like little living pets. If you need some bread, hey, yeast, they do the work for you. They fluff it up. But when they start eating sugar, they start producing carbon dioxide, so you can see some bubbling, which is kind of cool. They start creating that bubbly layer. You can watch them move under the microscope. You can give them some methylene blue, and some of them will eat it and poop it out, and other ones won't even go into because they're dead. So you can count how many living ones and dead ones just by giving it a little bit of blue dye. And the last, you just have gum medium for building slides. We already talked about the specimen dish hatchery, lens tissue. Already talked about that. You should have tweezers. Some people call them pickups, some people call them forceps, but tweezers, pickups, forceps, whatever. You should have a paintbrush, a pipette, or an eyedropper. I skipped over paintbrush, spatula. It's good for smearing. So if you need to, you have something that's thick and you need to smear it on your slide, you just drag and smear it. It's good for scooping too. What did I skip over? Pipette, got that. Pick probe. Yours probably has a blue cover on it. My blue cover fell off, but it's not that sharp. You should still be safe. Pencil, which we'll be using a lot of. Marker set, not by a lot of markers, but there's a marker with it. And a ruler that's in here. Oops. And a couple of labels in there. I'll just leave them together. They don't want to come apart. You have toothpicks, which I left out somewhere. I have one for representation. And these are really handy for using the stains so that you don't use the eyedropper. You just dip a little stain on it, and I'll show you how to do it later. But I would buy a box of toothpicks. They're like $1 for 500. You'll be safe forever, most likely. Pencil sharpener. I got it for you not as just a pencil sharpener. Yep, sharpens the pencil, but also because if you take a Phillips screwdriver, take that screw out, flip the blade over and put the screw back in, now it's a microtome. And a microtome is for cutting specimens. So like this, I'm not going to cut my finger, but if I take that apart, flip it over, I can take a carrot or an onion, and it slices, real thin slices of the carrot or the onion, so that I can take that slice out with my paintbrush. So the paintbrush will take a thin slice of something, pick it up and pull it out, spread it on my slide, stain it, and then look at it under the scope. Later in some of the add-on courses, I'll show you how to take like a bug, put it in wax, slice across here and get slices of the bug. So it's not just a pencil sharpener. Clear nail polish. So, clear nail polish is to help you mount things. You can take a slide, put a drop of nail polish on it, put a bug in it or whatever your specimen is, put another drop on top, and then make a sandwich with the cover sheet. So then you have the whole thing filled with nail polish that's going to harden and keep that thing sealed for at least a year, unless you bend it or break it. It's not as good as the permanent chemicals you can buy at a science shop, but it's good enough for what we're doing, and it's definitely good enough to help you see cool little things for short term. Clear tape. Okay. I'll show you how to use that. That's also useful for making slides when you don't have glass. Microscope cover. Already talked about that, to go over your scope when you're not using it. 3 AA batteries. Hopefully you have them in. And then the lab notebook guides. So this is your lab notebook on this side. Here's the guide on the other side, so that whatever you're doing, you can take notes on it over here. I know it feels like school when you take notes, but it doesn't have to. This is a place for you to jot down ideas or questions. I used to play music a lot, and I loved when you wake up and you've got this really cool idea in your head and you're like, oh man, I'm going to finish streaming and finish that idea, and I'm going to tell everybody about it tomorrow. And tomorrow comes and you're like, oh, I think I had a really cool dream. That's what your notebook's for. While you're in the middle of doing something, your mind's going to wander. Let the wandering mind write new questions or put new things down on that page. Don't let them slip away, because sure enough, you'll be focused on something else. And then when you go back to it, like, oh, totally forgot what it was. All right. Number 1 tool was your brain. Number 2 tool was your microscope or lab set. And then number 3 is keeping a notebook. Making sure you write down all those ideas, because those ideas are the golden. What if you're the person that figures out that bad breath is caused by a bacteria and the antibiotics cure the bacteria? What if you're a surfer and you're hanging out, swimming and on the beach, and you're noticing some algae and different living organisms that their temperature, when it changes, they start going through different cycles of life. And then you go back to your lab and you create a PCR machine that makes you a Nobel winning scientist. You know, you never know when you're going to come up with cool ideas. A notebook's nice to have so that when you're sitting around doing whatever, it's close to you. Da Vinci, one of my favorite scientists of all time. A lot of people say, well, he was an artist, he was an inventor. He was all of those. And when he died, he had 7,000 pages with over 7,000 illustrations full of notes. And when you look at his pages, they weren't just about the body on this page like you see in the picture. He would have little side notes around the corner about things that were totally unrelated that he wanted to think about later. Like his idea for putting egg whites in his paint to help it give a better shine. It was going to stay longer. So when he was painting the Last Supper, he mixed egg whites with all his paints, thinking it was going to be this perfect painting that would last forever. And it was the worst thing he could have done. So a lot of mistakes happen in your notebooks too, but that's the cool thing. You learn things that don't work. And that's what Thomas Edison said about inventing the light bulb. He tried over 1,000 different filaments before he came up with tungsten. Even cat intestine was one of them. And when he told people that he tried over 1,000 things, they're like, wow, so you failed 1,000 times. He said, no, I found 1,000 things that didn't work. So intentionally find things wrong. Think the way your brain wants to think. Your brain doesn't like linear. Your teacher probably hate me for that, but when you see an outline, your brain doesn't think outline. I mean, how many times have you sat down and tried to take notes and that outline just poured out perfectly and all of the 1s and A's and 2s and B's and all that lined up? This doesn't work like that. Your brain is meant to think in lots of directions at one time. So I remember teaching, when I was teaching at a nursing college, students always had, they were like, how do I study? What's the best way to study? And I don't know what the best way to study is, but I can tell you how your brain likes to study. And your brain likes to study in like not a fixed path. It will think of a word and come up with all the connections it can, which is really cool. So you might be working on something with bacteria, and then your brain goes off to some other microbe that you'd think was totally unrelated. But you've got such cool ideas, go with that pathway. So what I would tell the students to do is, here's how you approach a question. If you want to know a bunch of things about the muscle, and you know there's going to be a lot about muscle on the test, what you do is you write the word muscle down on a piece of paper and just go crazy. So if I write the word muscle down, actually, I did that last time, so I'm already prepared for that. Let's get something new organ, the eye. Okay, so you have that word like that, and then as fast as you can, you come up with all the connections you can. And so I might do cornea, lens, lateralis, that's the muscle. Pupil, ball, right, eyeball, lid, pad, oops, something got on my hand, hurt. And then that was like 10 seconds. So I came with 1, 2, 3, 4, 5, 6, 7 things because I write slowly. But what doesn't seem right? Did I do anything wrong? My mind wants to think of other things. You know, I may have gone to Steve Jobs after this, but yeah, I stuck with some of the things that are common sense to think of eye, but then like the iPad, what's that have to do with anything? But it's a creative thought. It's my brain going, hey, why do I have to be stuck in this little box? I want to think out of the box. So I would always tell students, if you can come up with the main topics, branch off like this, and then when you see a question on a test, look for the main word and just plug everything in, then look at the answers. If you plug things in and the answers, only one of those 4 has what you came up with in your head, stick with it. That's the way notebooks are meant to work. It's the way that notes are meant to be taken, is taking a thought and building on it. So if you get a chance and some random squirrel pops in your mind, run with it. Run with it in your notebook. That's my favorite thing to do. Right. Collecting, culturing experiments. We're not going to spend a lot of time on this right now, but when you need to prepare a specimen, it's not always going to be done in the time that you want it to be done. So sometimes you have to wait an hour or 2 hours or a day or 2 days. Some of the things that you're going to collect, you might have to prep. Like a lot of microorganisms, they like a wet environment, not drenched, soaked, but they like living in moist moss or the deeper parts of the grass where it's wet on the ground. And if you take that wetness away, they'll curl up in a ball and they'll dehydrate themselves until they get more water. So they kind of go in this hibernation state. In this section, I gave you some moss. If you open up the 101 envelope inside, I already pulled this stuff out. There's a paper towel. If you open it up, there's some moss in there. You can rehydrate it and take a peek at it later. All you're going to do, put a little piece of the moss, not the whole thing in there, with your tweezers. So with this one, I broke off a little bit of the leafy section, which is not leaves, it's moss, so it doesn't actually have a leaf, but everybody calls it leaves. And then this is a part of the root, which a moss doesn't have a root, but it looks like a root. It's called rhizoid. And I put it with just a little water, not to completely drown it, because these creatures don't like to be underwater. They like to be in moist environments. So bringing that water to go up and rehydrate the plant will hopefully rehydrate them and bring them back to life. So when you have a specimen, think about the environment at once, not just dump a bunch of water on it and see if it can swim. Think about what you're doing. Pond water is the same way. You don't want to dehydrate it. So when you're working with pond water underneath in the light, it's a bright light. It still generates heat, even if it's an LED. There's a lot of heat right in that spot where the light's focused, so it will dry out the water and you're going to kill your organisms. So just think ahead about how you're culturing or growing things. Heck, there's stuff in your refrigerator that's cultured, like yogurt. You know, somebody already did the job for you. Take a sample of yogurt and look at it underneath your microscope. Right? And then I just put more examples like hydrating moss or lichen off of a tree or soil. Teaspoon of soil with a little bit of water. Let it sit for a while and suck up some of the water and put it on a slide and see what you find. You might be amazed. All right. This is all about critical thinking. Keep your mind open. People 200 years ago thought that you got sick because of stinky air. I'm for real. If the air was stinky and you're like, oh, can't stand being here. They thought they were literally going to die because they smelled that bad air. It was called miasma. Because we didn't know anything about the invisible world back then. We didn't know about microbes. Now we know that a lot of the stinkiness is actually caused by bacteria and decay caused by bacteria and other microbes. Keep your mind open. Keep a critical mind. This little QR code goes to Dr. John Snow, who in London, he was probably one of the first credited epidemiologists. There was an outbreak of cholera back in the 1800s, and was it 1800s? Now I have to think about the date. But what's important is that he didn't believe people were saying that it was stinky air that was making people get diarrhea, vomiting, dehydrated until they die. Like literally they would vomit and have diarrhea until dead. That was cholera. What he did is he looked at all the places people were dying, so getting sick and then dying, and he looked at hot spots. And he looked to see if there was something central to those hot spots. Was it somewhere they were buying their groceries? Was it somewhere that they all socialized? Was it the local bar? What was it? Where were they hanging out, or what were they doing that they were getting sick? And so he looked at all the things people did regularly. Where did they go to the bathroom? Where did they get their water? Because back then you didn't have a faucet. You had to go to a central well, pump on the well handle. So you're pumping and pumping and pumping, and eventually some water comes out. You fill your bucket and you go home, right? And that's your water for the next day or two. What he found is that it wasn't a bar, and it wasn't like a food area where you buy your groceries and stuff. He found this well that he thought that most of the people were going to and getting their water out of, and thought it must be the well. The stink isn't coming from the well, but it must be the well. So he talked to the local church and said, because they own the well, can we take the handle off of the well so nobody can get water for a while and just see if anyone gets better? And back then, science and the church didn't go along, so it was kind of nice that they did get along, and the church took the pump handle off. And what they found is that all these people were angry because they couldn't get water out of that well, but there were other wells. So they went to the other wells, and sure enough, the people in that area, they stopped getting cholera. They started getting better, and they just left the pump handle off. And now it's actually like coated with gold or something that hangs at the church. But that was really the first epidemiologist, Dr. John Snow. And an epidemic is something that travels across the country. We're in a pandemic that's going across all the continents. But that's really the idea, is that if he had been stuck with it's got to be the stinkiness, he would just walk around with his shirt over his face all day. But he thought it's got to be something else. Be inquisitive. What's causing you to stink? There's a QR code down here that is a link to one of our STEM mini labs, where we talk to you about taking cheese out of the fridge, exposing it to different things, putting it in a bag, and seeing what kind of things grow on it. Be curious. Take a shower. Wash your hands really well, whatever. Take hand sanitizer, put it on your hands and feet, and then take cheese and put a slice on your foot, put a slice in your hand. Wait 30 minutes and put them in their own bags and see what grows. And what's even cool, you can see stuff on the surface grow on the cheese, but now with the microscope, you can actually see down at a microscopic level what's growing. Take a picture of it, give it to Google, and Google will tell you what kind of nasty stuff you have growing on your feet, right? You can test that. Cheese is a perfect petri dish, not artificial cheese like Kraft cheese, because that's so full of preservatives is not going to do anything, but real cheese. Get it exposed to some bacteria or fungus and it's going to grow like crazy. But keep a critical mind. Think of the invisible world as your new world. It's like a baby going to this new environment, wonder what's going to happen. What are you going to see down there? What are the kind of, if you look at pond water, or even some water from a little puddle in your yard, what kind of things are you going to see growing in there? If those things were life size for us, they'd be terrifying. It's crazy. But try things out. Next one, the complete history of microscopes. Not really, but enough for what we're doing. So microscopes have been around for a long, long time. Magnifying glasses even longer. Magnifying glasses, they started with probably a caveman taking a piece of quartz and polishing it with his bear fur from his kill, polishing it down until he could see through it. And maybe he was the first person to burn ants with a magnifying glass. You never know. But magnifying glasses were handy because, of course, they brought things closer. Until the Jensen family, father and son team, thought, what if we take two, right? If one lens is good, two's got to be better. So they kind of stacked them up so they could see through both. You could probably try that too. But you have two lenses. I didn't even think to try this before. Look at that, all the questions we're coming up with. But maybe you can increase the magnification with your lenses. Water lenses are not all that great. I see words. Ugh, see words. Nice. But give it a shot. See what you can do. Compound microscopes, they didn't really know what to do with it. They thought, hey, this is cool. We can see a lot of things. Robert Hooke came along and took a slice of cork, a tree slice, looked at it, went, wow, that's pretty interesting. He actually had 3 lenses in his microscope. He said, the shape that I can't see without the microscope looks like little cells in a monastery, like all the monks' rooms lined up, kind of like in a jail or a prison too. And so he called these little chambers cells. And we've used that word forever to identify the cells that build our body and build trees and plants and everything. So Robert Hooke started that, and we'll actually replicate his experiment. The next one was Anton van Leeuwenhoek, and he was actually a seamstress, and he sold drapes for a living. So what's interesting is that why was he playing with a microscope? It didn't make any sense, but he was fascinated with them. He loved polishing lenses. He was the best at polishing lenses, and little tiny ones. And then he put it in his microscope. It looked like, oh, you can see this book, like that. And if you look closely, there's a little piece of glass that sits on a flat plate, and there's one that's suspended. So he had kind of a compound microscope, but it was small. And then he would take a little drop of water and put it on a pan on the opposite side. And then he would hold it up towards the sun or a light. They didn't exactly have light switches back then, and the first picture I saw of him was him holding up to the sun, like, that's safe. That makes sense. But he could see something in that water drop moving around. And he said, like, it's like little molecules, but animals. So they must be animalcules that were moving around. And those were the first bacteria and germs that were recorded using microscopes. So he got a lot of fame and credit. He tested himself. He'd peel skin off and look at skin under the microscope, spit on it, body fluids he'd put on there. You just want to see everything. But I say that like it's a bad thing. It's not. It's really interesting. Just don't tell people about it because they're like, oh, crazy, right? Yeah, today I was looking at this bacteria I found in the corner of my bathroom. No, I didn't really do that, but I'm sure somebody would laugh at that. Right, types of scopes. So microscopes you're using today that you have actually from now on are compound microscopes. It's like the Janssen family built. So there are 2 lenses that are appropriately distanced so that you can magnify. You can go crazy. The 3 pictures here are actually electron microscopes. So instead of using light to send the image, they shoot electrons through the specimen. Your specimen is dead after that. It's not going to live through it. But this top picture is actually a cork cell, just one individual one. You can see the nucleus, that giant ball in the middle. The one below it is another group of cork cells, and the one below is the same group of cork cells, just closer. So it's mind-blowing how much detail you can see with an electron microscope. All of these fields of practice are things we're going to go through one at a time while you're playing and experimenting. They said 101s for us to just play with stuff, and I'll reference who uses a microscope in the real world and how maybe someday you can make some money doing it. Some of the jobs that use microscopes you might be surprised by, like textile scientists that use microscopes to look at fibers, like for dresses or tuxes or designer clothes, or coming up with the next cool kind of material, like making it out of banana or coconut hair for whatever. So they make a lot of money playing with a microscope and looking at cool little things all day. All right, next point, how vision works. Did I skip over something? Perfect. Cool. That means now we start getting our hands dirty. So how vision works, light works in 2 ways. Light's reflected, which means it bounces off of things, and it's also refracted. So reflecting light, if you see something in the room and it's, everything's like black and white here. If it's orange, this means that this thing hates orange, can't stand orange. When white light, like on the prism on the top of the picture, comes shining down, flip over here. That was totally not right. I work with it. Okay, well, great, and cut off my prism. So you can see the prism, the white light in your book goes through it and it splits it. So light is a wavelength. It's actually a lot of wavelengths when it's white. But when it goes into the prism, the refractive or the bouncing and bending properties of the glass as it goes through splits the wavelengths in different directions. When you look at something like this, the light comes down as white, it sucks in every color's wavelength except one, orange, and it bounces it, it reflects it back at you for you to see the color orange. So when you look at a tree and they're green and they're like, oh, trees love green. Isn't that cute? Trees hate green. That's why they send the green light at you. They bounce it off. They love all the colors but green, and they're sending that nasty green, that moldy looking green right at you. That's reflecting, that's bouncing. Refracting is when you're at the swimming pool with your buddies, right? And everybody's swimming, and you look at your friend, and you're standing up here, they're down here. Oops, I guess I have to turn this on for it to actually work for you. Hold on. Okay, like this. So water, you're up here, they're down in the water. When you're down there and you're looking down, oops, I get that perspective now, like that. The bottom, when you're standing above, the legs seem really short, the top of the body seems really long. Sometimes by looking at this angle, you can see it. But it's almost like the pencil's split. It's because of the refractive, the light bending properties of water. When the light goes through something, it passes through, but it gets changed. Nothing, even clear glass, isn't perfect. Things get changed as they go through. In this situation, the light gets bounced just enough, all the colors together, so it doesn't look like it's a different color. It just bounces it, right? Not bounces back, but bounces and turns it at an angle. That's refraction. Your eyeball, your microscope, do the same thing. Quartz, rocks that you can see straight through, plastics, whether it's a Ziploc bag or a piece of tape or whatever, all have refractive properties. If you can see through it, it's refractive. If you take a flashlight and put it in your hand and the light comes through and it comes out as red, it's still being refracted. It's being bounced off of the iron that's in your blood cells, and you're seeing that as a result.